Okay, today's topic is gene therapy. But before starting this topic, we should be knowing that we all are made up of genes, and genes makes us what we are. So what happens when these genes becomes defective or mutated? Obviously, there is a development of some disease. So to treat those diseases, which cannot be cured by the conventional therapeutic procedures like drugs, vaccines, or surgical procedures, we look for this gene therapy. So gene therapy is basically a therapy that involves the introduction of the functional genes into the cells to prevent or cure a wide range of diseases. This gene therapy, it presents a novel treatment method that uses genes or oligonucleotides as therapeutic molecule instead of the conventional drugs, and it targets both hereditary as well as the acquired genetic disorder. So basically, gene therapy, it is a technique that is used for correcting defective genes that are responsible for causing disease. Gene therapy targets either to the correction of the defective gene or suppression of its expression, suppression of the disease causing gene, or introduction of a normal healthy gene copy. Okay, so this gene therapy targets either correcting the defective gene that is present in the cell, or suppressing the disease causing gene, or introducing a normal healthy gene copy. Plus the epigenetic aspects of pathogenesis pathway, they are also covered. Epigenetic aspects means see, DNA is affected by the methylation patterns and histone modifications. All these processes that are going in the cell, they are also affecting the structure and the function of the genes. So it is not directly the gene itself, but the related mechanisms, they are also affecting the function of the gene. So this gene therapy covers those epigenetic aspects as well. So gene therapy, it involves the genetic disorder by inserting of a gene into the body in a functional or a physical sense. So when this gene is introduced into the cell of the patient, this process of introduction is known as gene delivery, and it occurs with the help of retroviruses generally. Apart from retroviruses, a number of other viruses are also being used as vector. For example, adenoviruses, adeno-associated viruses, and nowadays nanotechnology is also being used for the gene delivery. And okay, in humans, about 5,000 diseases, they are caused due to a single gene mutation. For example, cystic fibrosis, Tay-Sachs disease, Lesch-Nyhan syndrome, sickle cell anemia, Hunter syndrome, muscular dystrophy, plus a range of these kind of disorders like cancer, hypertension, atherosclerosis, neurodegenerative disorders. These all of these can be targeted by the gene therapy, but there are a number of criteria on the basis of which we select what kind of diseases we will be treating using this gene therapy. Okay, so first of all, the disease should be life threatening. The gene that is responsible for causing disease, we should be able to clone it. There need not to be a precise regulation of the gene once it is inside the cell, and there should be a suitable gene delivery system should be available. So now the next is research in gene therapy. So the real research in gene therapy, it started in 1960, but the first approved gene therapy experiment, it was conducted in September 1990 at NIH US under the direction of William French Anderson on a 4-year-old girl, Ashante De Silva, who was suffering from ADA, this is adenosine deaminase severe combined immunodeficiency syndrome. So it is a rare congenital genetic disorder of immune system, in which if this enzyme ADA, adenosine deaminase, if this enzyme is missing, due to which there is the T lymphocyte, they are not developed properly, and the immune system of the patient, that is crippled severely. So other diseases that are included for gene therapy, that are targeted for gene therapy are hemophilia, tyrosinemia, hyperglobulinemia, cystic fibrosis, cancers, etc. So the first product that was in the market was Glybera. So this was the first approved drug gene therapy product in 2013. Actually, it got approval in 2012. It cures this LPDL. This is lipoprotein lipase deficiency. It's a rare disorder, genetic disorder, and for this Glybera was developed. That got FDA approval in US. But the first gene therapy drug that was in the market, it was Genticine, Gendicine. It is a drug that targets cancer, and it got approval. It was developed by China, and it got approval in 2003. So China was the first country in the world that approved commercial gene therapy product, and the first product was Genticine. And in Western world, it was Glybera that got approval in 2012. So after this, next is after this brief introduction of gene therapy. So what is gene therapy? Gene therapy is the introduction of a functional gene to replace a defective gene in the cell. So this is gene therapy. It occurs with the help of a vector. The DNA, the functional gene, it is packaged into the vector, and this vector is introduced into the cell of the patient. So after, there are 4 approaches of gene therapy. These are gene replacement, gene silencing or gene interference, gene augmentation, and suicide genes. So these are the 4 basic approaches. Okay, first is gene replacement or correction. It is used to correct a defective gene to restore its function by homologous recombination. So in this figure, these are the cells, that is, they are the disease cells carrying the mutant gene, and this is the normal functional copy of the gene that is introduced in gene therapy. So these here it is showing the process of homologous recombination, in which this defective gene, it is replaced by the functional gene during homologous recombination, and this corrected gene that gets inserted into the genome of the cell, and now that these cells, they resume their, from disease they become normal, they resume their normal phenotype. So this is the gene correction method, the first method. Second approach is gene silencing or gene interference. It is used to treat cancers and other infectious diseases. It blocks the disease gene. How? I'll show you. So in this diagram you can see we have introduced the antisense gene. Antisense gene is the gene that is causing the disease. Its antisense gene, it is synthesized in the lab, and it is introduced into the disease forming cells. So when this antisense gene, it is introduced into the cell, it is going to code for RNA, and this disease cell is coding, it is also coding this diseased DNA, it is also coding for RNA. So this sense RNA, when it gets combined to the antisense RNA, and there is a formation of RNA duplex, so translation is blocked. Other pathway is that the antisense gene that you have introduced in the form of antisense RNA, and this RNA, it forms a duplex with the DNA disease causing gene, it forms a duplex with the disease causing gene, and this DNA RNA triplex is formed and transcription is blocked. So here translation is blocked and here transcription is blocked, and in both cases disease causing protein, they are not getting synthesized. So this is the targeted inhibition of gene expression by interference. Third approach is gene augmentation or gene addition. It cures autosomal recessive disorder, and in this, additional copies of genes, they are supplied into the cell. It occurs in the case, suppose a gene is missing or a gene is defective. Then we supply extra copies of gene into the cell to complement its function. Okay, this is the gene X. It is a functional form of gene. It is introduced into the cell that are deficient in this gene, and once the gene that gets into the cell, they resume their normal phenotype, and the corresponding protein starts getting expressed and normal phenotype is resumed. So this is gene augmentation, gene addition therapy. It occurs in the case where the gene, it is either deficient, it is missing, or mutated. Fourth approach is suicide gene. So this here the cell they undergo apoptosis, that is the programmed cell death. It is generally used, this approach is generally used in case of cancerous cells or to make the cancerous cells to prone to anti-cancer drugs. So in this approach there are two types, like it can be done in two ways. Number one, this toxin gene, it is put into the cell, disease cells, and these toxin cells they form the toxin protein, and the disease cells, they get killed by the expressed toxin protein. Second approach is this prodrug gene, it is introduced into the genome of these disease cells, and these cells they become prone to the drug. So when that patient carrying the cells in which prodrug gene is inserted already, they become more prone to the anti-cancerous or anti-proliferative drug, and they become killed when drug is used or when chemotherapy is used on the patient suffering from the cancer. So these are the four approaches that are used. Which approach we are going to use for gene therapy, that depends on the type of disease the patient is suffering from. So next is the type of gene therapy that we will cover in the next video.